Well, hi there. Welcome to part two of this discussion, this three-part lecture series on viruses, viroids, and prions, primarily viruses. Okay, we're going to look at viral taxonomy. So how are they named? What are we doing with their naming and classifying them? Well, family names end in viridae and genus names end in virus. So pretty simple. There's more to it than that, but these are the two that I wanted to just focus on. A viral species, then, is a group of viruses sharing the same genetic information and ecological niche, which would be the host. That's what its niche is. It would be its host because it has to live inside of a host cell. Common names are used for species, and subspecies are designated by a number. And virology is the study of viruses. So, for example, we have family Herpesviridae, genus herpes virus, subspecies human herpes virus 1, human herpes virus 2, 3, and so on. There are a few of them. Or family Retroviridae, lentivirus, human immunodeficiency virus 1, human immunodeficiency virus 2. So we have two of them. The coronavirus, we have SARS, coronavirus 1, which was there was an outbreak from 2002 to 2004. It wasn't a pandemic like we have this time around, but that was how that one was designated. SARS, Severe Acute Respiratory Syndrome, Coronavirus 1. And then today we're dealing with SARS-CoV-2, Severe Acute Respiratory Syndrome, Coronavirus 2. And that's its designation. So it's a subspecies. So we have coronavirus 1 and we have coronavirus 2, which is the one, of course, that's causing the global pandemic that we are living in at this time. So this is how they're designated subspecies, and this is just a snippet of it. There's a fuller taxonomic classification for every virus, larger than this, more extensive. But I just wanted to show you the very end of it here, of their classification, how we get down to the subspecies level. Growing viruses are problematic because viruses must be grown in living cells. So it's not like with bacteria we can just slap them on a petri plate, incubate them, and then investigate. Viruses are problematic because you have to have living cells, but when living cells become infected with the viruses, they die. So there's nothing left then for us to look at or to research with. Most of what we know of viruses is from bacteriophages because they're easily grown on bacterial cultures. So what you're looking at here would be a petri plate with a lawn of bacteria. That would be the plate is covered with it. And all those little circles are what are called plaques. And that's where the bacteriophages have killed the bacteria. Remember, the phages are the viruses, and bacteriophages is telling us that they are well, let's look at the word bacteria. Well, obviously bacteria. Phage comes from the Greek word that means to eat. So I guess you could say they're eating the bacteria. So these are viruses that are killing them. And that's what those little circles are. Those are what are called plaques. And so that's where a lot of our research has come from, because we can obviously grow bacteria like crazy and they replicate very quickly and rapidly. And so we get a lot of research done using bacteriophages. However, there's a cell line that I'm going to talk about here in a minute, in which we're using actually eukaryotic cells, human cells. Growing viruses, so we can get animal viruses. They may be cultured in living animals, and that becomes a little bit problematic ethically, because the animals, they die, obviously, from the research with these viruses. And then we examine the deceased mouse or rat or whatever it happens to be in embryonated eggs, like you see in this picture here, or in cell cultures. With embryonated eggs, meaning an egg that has been fertilized, a hen's egg has been fertilized, so the rooster has been in the hen house. What happens then is at a certain point in the development of this embryonic chick, little holes are drilled into the eggshell, and through that, needles are inserted, and the egg at its different membranes are injected. They're inoculated. And viral growth is signaled by the death or damage of the embryo or lesions on the egg membranes. And so why do we use embryonated ones? Because we get a much greater variety of membranes as a result of the fertilized egg. Rather than if it's an unfertilized chicken egg, you don't get the membranes, you don't get, obviously, the embryo developing inside of it. And so this is why a person may be asked if he or she is allergic to eggs prior to vaccination, and that's going to the wayside because this type of growing viruses is not really used all that much anymore. The number one way that they're grown are in cell cultures. This is the most common way to culture viruses today. Normal viral infected cells will show cytopathic effects. Okay, what's a cytopathic effect? Cyto means cell, pathic means bad, death, something related to that. So they're going to show cell death or bad stuff to a cell effects. So viruses will cause different things, a variety of things to happen to cells whenever these cells become infected with the viruses, which we'll talk about in a few minutes. However, what happens is they die out after just a few generations. So we have these cells and we're infecting them with viruses and it's going good for a while, but then the cell line's gone. It's done. So what do we do? Well, we can get continuous viral infected cell lines from transformed or cancerous cells that can be maintained indefinitely. And the cells that we use by far are called HeLa cells. And what you see here in the graphic down below, so you get tissue treated with enzymes to separate the cells. The cells are suspended in culture medium. The normal cells, they grow in a monolayer across the glass or the plastic container, whatever they happen to be in. And the transformed cells are continuous cell cultures. They don't grow in a monolayer, they pile up because they're cancerous. Remember, there's no contact inhibition. Their cell cycle is the pedal to the metal, and the cells are just dividing and dividing and dividing. Normal cells ignored, transformed cells, they're the ones that are researched. Okay, back to HeLa cells, named after a woman whose first name was Henrietta, that's where the HE comes from, and her last name was Lacks, Lacks, Lacks. LA is for her last name, so HE for her first name, Henrietta, LA for her last name, Lacks. So Henrietta, and she was lived in Baltimore, and in 1951, so she was 30. So she was born, I think, 1920. She died at 31, so she was young. She went to Johns Hopkins Hospital in 1951 to have this. She was bleeding a lot, and it was determined that she had cervical cancer by the doctor there. His name was Howard Jones, Dr. Howard Jones at Johns Hopkins University, which was one of the few places in the country where an African American could be treated. Some of our sad history of racism and prejudice against African Americans. But Johns Hopkins was one of the few places where an African American could receive treatment. So Henrietta goes to Johns Hopkins, and the doctor, Howard Jones, he biopsies part of the tumorous part of her cervix that was growing. And he took it from her, and she didn't know it. And he had seen that it was unlike any other cell that he had ever seen, and any other group of cells. They were reproducing like crazy. Like their numbers were doubling in 20 to 24 hours, which is unheard of for human cells. They just don't do that. And it kept going and going and going. Well, he was a physician, not a scientist, so he sends. Remember, he's at Johns Hopkins, which was not only a hospital, but it's a research institute as well. He sends them to a virologist. His name was Dr. George Guy, and he looks at these transformed cells. He had never seen anything like them either. So he was doing research on viruses and cancer cells, but eventually normal cancer cells would die out. They became infected with the viruses, and they did better than normal or non-cancerous cells, but eventually they die out, and it was like, oh man, why'd you have to die out so soon? So they lose their research cells. Well, Henrietta cells never died. They were infected with the viruses, and they would replicate and divide and replicate and divide, and they became extremely useful cells. There were no cells like them on Earth anywhere, and there haven't been any cells like hers since then either. Her cells are still alive. They're around the world. There are bajillions and bajillions of them around the world today. And there was a book written by an author, Rebecca Skloot, and it was called The Immortal Life of Henrietta Lacks. So she was an author. She found out about the Henrietta story, and she published a book in early 2000s. Should have looked up the date. Sorry. about Henrietta and her cells. So she did a whole bunch of research, met with the family, wrote this book, got it published. Oprah Winfrey, she read it, was very moved by it, decided to fund a movie on HBO, The Immortal Life of Henrietta Lacks, which was released in 2017. And it gives Henrietta's story. It was a problematic story because Johns Hopkins took the cells, at least Dr. Jones did. He took the cells, and Henrietta did not know about it, nor did anybody in her family. They took it, they researched with it, and George Guy, the scientist from Johns Hopkins, he then sent the cells around the world, and scientists around the world began researching with Henrietta's cells. They fueled all kinds of scientific research. There was a whole lot of money that was being put into the research of these cells. Universities were researching with them. Cancer institutes were researching with them. Labs around the world were researching with Henrietta's cells. And in fact, there was such a demand for her cells that at least one facility started pumping out trillions of her cells every day and then sending them off. And they were making money from it. But Henrietta's family did not see a penny from it. So this became an ethically problematic situation. Whenever it was publicized by Rebecca Skloot in her book, The Immortal Life of Henrietta Lacks, there was a lot of attention that was given to it. Johns Hopkins then in 2010 decided that they would offer a public apology, and you can go on their website and you can see what they have done in order to support the Lacks family, seminars, attention, money, donations, contributions, and I'm sure they did a lot to publicize or even contribute to Oprah Winfrey's movie that she produced to get this message out. So anyway, yeah, there is a little bit of difficulty with the background of that story because, and so, but from it now we have, informed practices now. Patients cannot have tissue removed from them unknowingly without their consent, without their permission, and so forth, all because of this. In 1951, it wasn't an issue. There were no regulations or laws against it. But now from this, we've learned that, yeah, there needs to be, along with other situations as well. But anyway, HeLa cells, yep, they're still alive, and that's why she titled it The Immortal Life, because I guess she kind of is immortal. Her cells are alive and will continue to be alive around the world, and that's a hyperlink to a good TED Ed video on HeLa cells. The Johns Hopkins website also has a video as well on YouTube to promote this story too that you might want to check out as well. Good explanation of Henrietta and the story behind it. Interesting story. How do we identify them? Well, I talked about using spikes and so forth, but sometimes that's difficult because they're so small, and so how are we able to see them? So we can use indirect methods. One of them are called CPEs, or cytopathic effects, cell deterioration caused by a virus, and viruses typically have characteristic CPEs, meaning this virus seems to have this type of cytopathic effect, whereas this virus has a very different cytopathic effect. So we can identify them by the damage that they do to their host cells. Serological tests, which can identify a virus by its reaction with antibodies, so there are a number of serological tests that can be used, or just identifying their nucleic acids, running their genome and doing ELISA tests or PCRs, polymerase chain reactions, and identifying their nucleic acid sequences, looking at the RNA sequences, the introns that we look at. Those are ones that segments of RNA that we look at as well to identify these viruses. This is what some cytopathic effects look like. On the left there, those are epithelial cells, simple squamous epithelial cells, and the one at the top, middle, that's a normal squamous epithelial cell, but down below you can see how the cells clump up, and those little round circles, those spherical objects, those are nuclei, so it causes multinucleation within the cells. and it gives them this funky appearance. It's unusual. It's a cytopathic effect. That's not normal. Why multinucleation? More nuclei, more nucleic acids available to the viruses so that they can make more of themselves. You can see in the bottom picture on the left, that's normal tissue under the microscope, and on the right, same tissue infected with viruses. You can just tell if it doesn't look quite right, does it? So these are cytopathic effects. There are two ways that bacteriophages will multiply, and that's through the lytic cycle and the lysogenic cycle. We'll look at the lytic cycle first. So remember, the virus must invade and take over a host cell in order to multiply. It needs the enzymes, it needs the nucleic acids because they lack those enzymes and nucleic acids. They have the basics, but they don't have all that they need. They don't have all the essentials. And so that's what makes them, remember, obligate intracellular parasites. They have to be inside of a cell, and they parasitize the cell. They take stuff from it at the expense of the host cell. So there are 5 basic steps to the lytic cycle. APBMR. APBMR, attachment, penetration, biosynthesis, maturation, and release. Attachment is self-explanatory. The phage, remember we're talking about bacteriophages infecting bacteria. We'll talk about animal viruses later in this chapter. The phage attaches by tail receptors to the host cell receptors. Then they penetrate. Phage, lysozyme, which is an enzyme, opens the cell wall, puts a little tiny hole in it, DNA gets injected into the cell. Now this hole is not large enough that it causes the bacteria to burst. It's a very, very small hole. Remember, viruses, especially bacteriophages, are tiny. And so the hole that's penetrated through the cell wall membrane of the bacterial cell is so small it does not cause it to burst. So bacterial cell, okay. Biosynthesis. So now it's going to start making stuff. Production of phage, DNA, and proteins. No complete phages yet, but they're starting to make the bits and pieces. And it's now is when the host cell has been taken over, because it's using the nucleic acids in the host cell to make more of themselves, or more pieces of itself. Maturation. Now we put the pieces together like a puzzle. We're building a Lego here, bit by bit, and now it's assembled. Then there's release. Phage lysozyme breaks the cell wall. And remember, there are thousands upon thousands of these new baby phages inside the bacterial cell. So whenever they release their enzyme, and a whole mess of them release their enzyme, that's the end of the bacterial cell. It bursts, and now the babies are released to wreak more havoc. So in picture form, it's like kindergarten, pictures are very helpful. Even as adults, of course, we like to see pictures as well. So you have the attachment where the bacteriophage attaches itself to the bacterial cell. Penetration, the DNA gets squirted into the bacterial cell. Biosynthesis, new bits and pieces are made. Then we have maturation and release. The bits and pieces are put together, assembling the bacteriophages, and then release. Whee! Out they go, out of the bacterial cell that has burst. And so this is why it's called the lytic cycle, because the cell bursts. And remember that bacteriophage video I told you about by Kyrgyzstan? Here it is. So my college students, look at the PowerPoint, click on this hyperlink, and if you want to learn more about bacteriophages, that's a pretty decent video to watch. This is it's a basic one-step growth curve associated with the lytic cycle. So what happens is here, you start out with, on the y-axis, you have the number of infective phage particles. So we start with 1 on the far left. They penetrate, we go down to 0. So there are none left. Then it takes about 20 minutes to undergo biosynthesis and maturation. So they're making their bits and pieces, they're assembling them, and then at about 25 minutes, boom, that's what we call the burst time. Phage particles are released by lysis, and they all get released, and they all go out and infect other bacterial cells. There's a 0 population from minutes 5 to 25 because the viruses are taken apart in the cell. That's what's called the eclipse period. So we can't count them because they're all just, they're maturing. There's bits and pieces. There's not whole phages that are in there. So we have a 0 population count. And this is a kind of an actual time. It's an average, but it's not just like, whoever made this graph thought, we'll just stick with minutes 5 to 25. We'll make that biosynthesis maturation, whatever. Now, I checked it out, and yeah, this is a typical one-step growth curve. It's called one step because you get penetration and then boom, it grows from there. Okay, that's the end of part 2 of this 3-part lecture series on viruses, viroids, and prions. Please stay tuned for part 3, and thank you for watching.